So today's lesson we're going to look at genetic drift and gene flow, which is basically two other ways that an allele. We looked at. Uh, just going to be mentioning this again in other slides anyway. But before I talk about the other two, let's see if you guys still remember that. So this image below depicts the changes in a population of mussels over time. In the image, the selection pressure is what? What do you see as the selection pressure acting on this population? Can I get Janita? Right? No, you're right. The bird, right? The predator is the selection pressure. Basically, create basically a pressure that's selecting for a specific phenotype within the population. Okay, and that's the first part, right? Selection pressure is and then the population that selection is acting against is. Can I get Melinda? What part of the population is the selection acting against? That's what they call that color. I don't know. Mint color, yes. The mint color muscles, they're being selected against. And the advantageous phenotype is, can I get Abby? The gray color, right? So this is an example of what form of change in alleles. Sorry, what is the name of this overall process? Can I get Zoe? Yeah, natural selection, right? So there we are. That is basically one of the examples. So if I have to define natural selection, remember that is a mechanism of evolution in which the individuals that are best and that basically that have an advantageous phenotype in that specific selection pressure will be able to pass on their alleles. And this is just one of the mechanisms in which a allele frequency within a population can change. The first one we looked at was mutations, and that's when a mutation randomly changes the genotype within a population to change the allele frequency. The second one is natural selection, where a selection pressure is acting on a population and then selecting for a advantageous phenotype with a selective advantage, right? So what are the four requirements of natural selection? Can give me all of them. Jasmine, what are the four things you need to have within a population for natural selection to occur? Variation, one. That's the last one. You're missing two in between. Selection pressure, that's the second one. Selective advantage, and then the last one, which is heritability. All right, well done. Exactly spot on. Okay. That is not arranged properly as well. All right. Okay, let's look at this question. A population of lizard species, species name, are found on the main island of the Bahamas. There is natural variation between the phenotypes of individuals within a population. Explain how natural variation can exist between individuals within a population. So how is it that variation can exist within a population? What is allowing for the variation to exist? So remember that variation is the phenotype, right? The expression of a gene makeup. So if you're thinking about what is the cause of what is causing that variation to exist, you can always go back to the source. What allows the phenotype to occur? The genotype. Remember that phenotypes are the physical expression of a gene makeup. So when you're thinking about what is causing that natural variation, you can always go back to the fact that their genotypes are such that they have created different allele combinations through sexual reproduction, thus allowing them to have natural variation. Another one is the first method of allele frequency change, which is mutation. Okay, this question is specifically very hard, as you can see. The 3-mark question, only 1% of those who did the paper managed to get 3 marks. 50% didn't even get a mark. So you need to be able to understand what are the ways in which allele frequencies can alter, meaning how can there exist natural variation. One is the different ways that allele frequencies can change, mutation, or the today's topics that we're going to look at. You can also use those as answers, but best ones would be something like how the genotype is made up of different allele combinations in sexual reproduction, allowing for natural variation to exist. Right? Like there's blood in my mouth. All right. So like I said, right, what are the two ways that allele frequencies are altering within a population? So far, we've looked at mutations and natural selection. That's the natural processes in which a allele frequency within a population can alter. And how does it that mutations can contribute to evolution? Because mutations, specifically, what type of mutations can be passed on to the next generation? Can I get, sir? Yeah, what type of mutation can be passed on to the next generation? So there's two types of mutation. Those are the examples of mutations, right? But these, when I talk about what type of mutation can be passed on, remember that if a mutation that happens on your skin, is that going to be passed on to the next generation? Not really, because that is a somatic mutation. Yeah, exactly. Germline mutation is the kind of mutation that happens to your reproductive cells. Those are the ones we can pass on to the next generation, because basically those reproductive cells end up making a new life, so they will carry on those mutations. Whereas any mutation that happens to your somatic cells are going to stay with you and won't be passed on necessarily, right? So yeah, like I said, these are the two mechanisms of evolution that we've looked at so far. Today, we're going to be looking at two more mechanisms of evolution. In other words, two more ways allele frequencies can alter within a population. So the two ways that can happen is genetic drift and gene flow. Like I said, the study point is that you guys will look at how changing allele frequencies in a population can be caused either by natural selection, genetic drift, gene flow, or mutations. Okay, they're the four main ways that allele frequency within a population can change, and they are also the four ways that evolution can occur. So they're considered as the mechanisms of evolution, because what they're doing is that they're changing the allele frequencies within a population going towards one direction, and eventually that can actually lead to evolution occurring over a long period of time. Okay. Yeah. So we're going to look at what are the biological consequences of these two forms of two mechanisms of evolution as well, and we're going to define what they are and how they work. Okay. So keep going back to the same thing. So far we've looked at this is mutation, right? Just a short recap. Like I said, mutations can randomly change the allele frequencies within a mutation such that there's emergence of new alleles, right? So you could have an entire homozygous recessive mutation, and they reproduce to produce more homozygous recessive. But then due to a random mutation, you can suddenly have a heterozygous individual within that population. That is how mutations can lead to a change in the allele frequencies. When we think about natural selection, this is when basically a selection pressure is selecting for a advantageous phenotype within a population, and over time, due to the advantageous phenotype having higher fitness, meaning higher survivability and reproducibility, they will start to take over the population, the allele frequencies, and that's how we can see it here, right? At first, you start off with a balanced population where there's a high variability between light brown fur and dark fur, and then due to a selection pressure targeted by predators that select against dark fur, they will select for the light brown fur, meaning that more of the light brown fur will be able to reproduce into the next generation, reducing the allele frequency of the dark fur and increasing the allele frequency of the light fur. And you can even see that reflected in the allele frequencies, where if the allele responsible for the light fur is the dominant allele, and the allele responsible for the dark fur is the recessive allele, you can see how the allele frequency changes to reflect that as well. So the dominant allele will be more increase in terms of frequency over the generations due to it being selected for, and over time that changes the allele frequency of the entire population. So that's the two ones we've looked at today. I mean, in the previous lessons, today we're looking at the two other ways allele frequencies are changing, which drive evolution, which is gene flow and genetic drift. So the first one is, what is gene flow? So gene flow is basically the flow of alleles between different populations due to migration or interbreeding. When it comes to interbreeding, that is different from inbreeding, because interbreeding is basically talking about when the same species between two different populations are breeding, whereas inbreeding is when it's the same population that are breeding over and over, right? So interbreeding is different from inbreeding. Make sure you guys are aware of that. And when I talk about migration, that's specifically immigration and emigration, right? Make sure you guys know the difference. Immigration is what's the word? Is the introduction of alleles within a population, and emigration is the removal or exit of alleles within out of a population, right? So that's the easiest way. I always remember them. Basically, immigration is introduction, emigration is exit. So that's always helped me remember them as being that separate words. So why can this happen? It's usually due to the fact that these populations, two different populations, are close proximity within each other. So one individual from this population suddenly emerges in that population, or otherwise, or due to natural forces that are removing any barriers between them. So let's go into a bit more into this, right? What is immigration and what is emigration, and how that actually influences what the allele frequencies will look like after that, right? So immigration is when a new individual, a new organism has moved into a population. So essentially, they are introducing new alleles to a population. And as we've looked at before, when you're introducing new alleles in a population, you are increasing the genetic diversity of that population, because suddenly they have alleles that they wouldn't have had if that individual or organism had not come in, right? For example, look here, right? You see the purple color beetle, and due to the fact that that purple beetle has immigrated into the population of brownish beetles, there is suddenly an increase in the genetic diversity of that population. So that population is actually better off due to the increase in the genetic diversity, because as we've looked at in the natural selection chapter, whenever there is an increase in genetic variability, natural selection, they can more likely survive any selection pressures that act on them. If there is a low amount of genetic diversity, then a selection pressure can actually lead to the extinction of that population. So this kind of change is usually very meaningful to small populations, right? Imagine in this population, you can really see a change because there's a very small amount of beetles and there's low genetic diversity in that small population. So when there's suddenly one immigration of purple beetle, there is a change in the allele frequency that's significant within that population. But imagine if there's like 1,000 beetles, when then a single beetle has joined into that population, the effect is not as prominent because there's already a huge number of beetles within that population, which would lead to it having a already high amount of genetic diversity. So when we're talking about genetic drift and gene flow, they both are basically going to have a high impact in low populations and relatively low impact in high populations due to the fact of low populations, their allele frequencies can highly change due to the insertion of removal of any alleles. So that's for immigration, where new alleles are being added in. What about for emigration? So like I said, immigration is when something is coming in. Emigration is when something is exiting the population. And as always, if alleles are leaving a population, that means that genetic diversity is going to be generally decreasing. Because when, let's say you have a small population of geese like this, where there's a mix of brown geese and also white geese, suddenly one of the white geese or two of the white geese all start to immigrate to a different population. That in turn decreases the genetic diversity of the population that it left from, right? So if a new individual is coming into the population, it increases genetic diversity. If an individual is leaving a population, that reduces overall genetic diversity. It's not that hard to link those ideas together, right? So, like I said, as with a small population, this in a small population, this can be very detrimental on the genetic diversity of a population, because imagine if all three of these geese decide to emigrate out of this population, suddenly all that's left is the brown geese, which greatly which is going to be very detrimental to the population in terms of its survivability. If it encounters a selection pressure that selects against brown geese, then that population altogether can become extinct. All right. Anyone confused so far? Okay. So basically, if something is coming into the population, genetic diversity increases. If something is leaving the population, the genetic diversity of that population is decreasing. All right, moving on. So how is it that migration and interbreeding can be connected? Basically, we have two examples here. Both are both are emigrating from that population into the other population. So the red one is moving in here, the blue one is moving in there. Okay, once they've moved into their respective the different populations, they are going to be breeding with the populations from that specific they're going to be breeding with those specific population. So that's what you refer to as interbreeding. When an organism from population A moves to population B and breeds with one of the organisms there, and vice versa, right? They are basically breeding with a population that they are not a part of originally. That in turn will carry unique alleles and increase the genetic diversity within that population, right? So for example, let's look at this bluebird, right? Once this bluebird immigrated to the population B, it will breed with the birds in that population. And you can see that the alleles for this population, right? So basically, the alleles for the bluebirds are homozygous dominant for HH. And when they move into this population, which has a homozygous recessive allele, you can see that the offspring of the immigrated bird will suddenly have the genotype of heterozygous capital H and lowercase h, which is in turn saying that the genetic diversity is increasing. So essentially, interbreeding is, individuals carrying unique alleles interbreed and will act to increase the genetic diversity of the gene pool. Essentially, think about this. How did Australia look like about 100 years ago? The population was leaning towards a very high Caucasian majority, right? But today's population of Australia is much more diverse due to the fact that more and more populations, more and more humans from different populations have migrated here and interbreeded to create higher genetic diversity, right? That's why humans are actually a good study of population genetics, because we are basically modeling it ourselves, right? Genetic diversity has increased due to the fact that different populations are interbreeding. Again, these words are very weird to use when we're talking about humans, but it is still applicable in that sense. All right. So that is gene flow, right? Gene flow talking about migration and emigration. Don't have to overthink it. Immigration is coming into the population. Emigration is leaving the population. And when new organisms come into a population, they will most likely interbreed, allowing for an increase in genetic diversity as well. Okay. So next one we're looking at is genetic diversity, right? This is when there is a sudden change in the allele frequencies within a population due to a random and sudden occurrence, right? This, like I said, with gene flow and genetic diversity, this is going to be a much more pronounced within small populations where it's going to be a much more higher effect, right? So how is this different from what are the two examples of this is that the bottleneck effect and the founder effect. Basically, these are two ways that the population's allele frequencies are going to randomly and suddenly change in a very drastic effect, and that can be considered as genetic drift. So under genetic drift, the two common occurrences are the bottleneck effect and the founder effect. Both of them are going to see a random and sudden event that causes the population's allele frequencies to very drastically change, right? So, like I said, how is this affected by population size? Within a big population, they can undergo genetic drift and still retain a lot of their allele frequencies originally. Not to say retain, but they can still have diversity within that allele frequency, and that allows them to have survivability. But within a small population, that sudden loss of alleles can actually have a very detrimental effect onto their population sizes. So we're going to look at the specific examples of each of them and how it impacts on the survivability. Don't get too confused yet. Okay, so let's look at this. Right, the bottleneck. This large portion of the population is wiped out by a random event such as a natural disaster or even hunters. Right. So let's look at this example up here. So we start off with a population of seals that has a amount of genetic diversity, right? We have gray seals, we have brown seals, white seals, and black seals, and they all are having within the same population, and there's a high amount of genetic diversity within this population. But suddenly there is a random occurrence that changes this, where the hunting of seals in the late 1800s greatly reduced their population size. So how is this actually different to natural selection and the selection pressure we talked about? Is that selection pressure is selecting for a specific trait, a specific phenotype. Whereas with genetic drift, it's going to be random. It's going to be unclear which phenotype is even, there is no phenotype that is being selected for. It's randomly annihilating a bunch of the population, leaving behind just a few individuals from the original population left, which is why it's considered as random and unpredictable, because with selection pressures, one phenotype is selected for, whereas with this, there's no phenotype that's being selected for. It's just suddenly the population is being wiped out for no reason, leaving behind only a few individuals, and they are gonna impact the allele frequency beyond that. So you can see this, right? Think about it in terms of the seals that were hunted in the 1800s. Their numbers were greatly reduced, leaving behind only about the surviving population, and thus the allele frequency has greatly changed from the original one, because whatever genetic diversity that used to exist changed a lot due to the random hunting of seals, leaving behind only those that survived to those who survived left in the population, and whatever is left, that's going to determine the allele frequency of the population in the future as well. Because due to the loss of those alleles in the start, the alleles responsible for gray seals or black seals, due to the loss of those alleles, whatever's left is the alleles responsible for brown seals and white seals, and only those alleles are going to be passed on moving forward, because these alleles were hunted for and they were lost. And sorry, they were hunted and they were lost, leaving behind whatever's remaining to carry on the population from there. Yes.
Or consider tasks, but all choose tasks. So is that genetic drift, bottleneck effect, or selection?
That would be a selection pressure, because they are selected for the, yeah. So because when seals were hunted, they didn't care what color the seals were. They were just randomly hunting the seals for the meat. And that is why it's considered as a genetic drift, specifically the causing a bottleneck effect, because it was random. But in the case of hunters wanting to find elephants with long tusks, they are selecting against elephants with long tusks, and they're selecting for elephants with short tusks or no tusks at all, right? So selection shift could be also natural disasters. Let's say a natural disaster, but then usually a predator is involved or some kind of hunting is involved, because something is selecting actively for that population, that phenotype. That is usually caused more often by hunters or predators, but they can also be supported by natural disasters. For example, think if a volcano happened, there's going to be a lot of ash produced. So whatever animal can use that ash to camouflage itself will be able to have a higher selective advantage compared to the same animal that couldn't camouflage itself, right? So let's see. Think of a moth that has two colors, white color and black color. The black color moth will be able to hide itself better in a ash environment compared to a white moth. So the white moth would be selected against because the predators can still see them and hunt for them, whereas the moths with the black skin will be better suited for that environment. So it will be selected for. Okay. I know this is a very complex idea to try to bridge together, but all you need to remember right now is that with natural selection, there is something specific phenotype that is selected for, right? But with the genetic drift, there is a random and unpredictable event that is causing the change within the element frequencies and how that impacts the population moving forward. So here, because the seals were hunted for their meat, and indiscriminately, whatever remaining seals, they start to influence the allele frequency of the population moving forward. So only brown and white alleles were passed on to the future generations, and only those alleles can be found further down the generations, as you can see in that picture, right? So, like I said, different from selection pressure, because this is random and not selecting for any phenotype. It is just eradicating a bunch of the population. And why is that a horrible thing? Is that any unique alleles that were present in that population could most potentially be wiped out, losing out on a lot of genetic diversity in the population. So as you can see in this picture diagram, right? At first, there was a lot of genetic diversity in the colors of the seals, and then suddenly, due to hunters going for all the seals, whatever's left is the brown and the white alleles. Sorry, what brown and white seals, and only they can make up the population moving forward. And comparing this population to the one originally, you can see how the genetic diversity has changed. The original population had a lot of genetic diversity, whereas this one only has basically has a reduced amount of genetic diversity. All right. Anyone still not getting this one? Okay, moving to the next one. The next one is founder's effect. So this is different from the bottleneck effect, because the bottleneck effect is talking about a random event that wiped out a a large portion of the population being wiped out due to a natural random event, like a natural disaster or something like that, or hunters. So there's a bunch of factors that can be causing this. Basically, anything that causes a large portion of the population to be randomly wiped out. So it's specifically randomly wiped out because it's not like any of them was selected for or against. It's just randomly wiping out a portion of the population, leaving behind whatever's left to basically reproduce and restore the population to what it was. With a founder effect, however, what's happening is that a small number of individuals from one population basically starting their own population separate from the original population. So this is your parent population. It has orange and gray butterflies. Suddenly, a few of these butterflies separate from the original population and make up a new population themselves. So what happens is, if, let's say, in this top example, 3 butterflies went to this population and all 3 of them were orange. So what happens is any other butterflies that emerge from that population of 3 orange butterflies will also only be orange, right? Here, it's more representative of the original population where some of the gray butterflies and also the orange butterflies moved into this population. So you can see the original population being represented here. Whereas in this population, again, only the gray butterflies moved into this population. So any other organisms that are going, any other butterflies that emerge from this population is just going to be the gray butterflies because they have basically influenced the future generation where a new population is reproductively isolated from the original one. This in turn creates unrepresentative samples of the original population. So in the original population, there were gray and orange butterflies, but in these 2 populations, you can see due to the founder effect, there is no, they are losing basically one of the original colors that the populations had. So essentially, this also leads to a lower genetic diversity in the new population compared to the species as a whole, due to the fact that whatever original variation that they had, they've lost it by forming a population with only a specific phenotype, right? So we start off with a bunch of different colored beetles, as you can see here. And due to the fact that these 3 beetles separated from that population, and then they formed a new population, every time that they breed to create a new generation, they'll ensure that it becomes a very homozygous population with the same alleles. And it won't have a high amount of genetic diversity as a result. So this also leads to a lower amount of genetic diversity in the new population compared to the species as a whole. Anyone confused by this? So this can usually be caused by natural disasters. So more often than not, any change in alleles can be caused by, any change in terms of genetic drift or gene flow can also be caused by natural disasters or environmental factors that basically causes changes within the population, either to make them immigrate or emigrate, or for them to be wiped out to become into these kind of effects, right? So it can either cause a bottleneck effect where whatever's left, the original population is forced to reproduce and restore the population to what it was, losing the original diversity that it had, or it can be due to the fact that a small portion of the population separated from the main population and had to basically re-emerge with whatever it had, losing the original diversity it had in the first population. All right. Both examples of genetic drift often lead to a reduction in genetic diversity of the population, where basically the bottleneck effect will reduce genetic diversity through the removal of alleles randomly and unpredictably, it will remove a bunch of alleles from the original population, leaving behind whatever allele that is left over to just reproduce and become prominent within that population. With the founder effect, it reduces genetic diversity through the establishment of a new population with a small unrepresentative sample from the original population. Right? Basically, if only blue beetles separated out from the original population and reproduced, only blue beetles will emerge from that population, causing it to basically only have a high amount of that allele present in the population. So we can look at this and then we can model it with either the bottleneck effect or the founder effect. Let's try doing it with the bottleneck effect first, right? So this is a population of beetles. There's a high amount of genetic diversity that is present. There's orange beetles, there's green beetles, there's black beetles, and then there's blue beetles. So suddenly a random event has occurred which wiped out a big portion of the beetles, right? Maybe they suddenly lacked food to so suddenly were lacking in a lot of resources, meaning that they couldn't find food, so a bunch of the beetles started dying off. And whatever was left was these three beetles, right? These three beetles where two are blue and one is orange. So it has already lost a bunch of the genetic diversity that it originally had. And these three beetles will restore the population to its original numbers, but due to the fact that it had lost the original genetic diversity, the population that comes forward from that will have a low amount of genetic diversity, right? They are basically only reproducing to carry forward these alleles to the next generation, losing that original allele diversity that it had at first. That is for the bottleneck effect. But now let's look at the founder's effect in this example. So let's say there's this population of beetles. They are high in genetic diversity, but due to some random event, three beetles were kicked out of this population, causing them to separate out and create their own population. Maybe there was a natural disaster that created a barrier between these three beetles and the original population, or some other event that has occurred to basically separate them out of the original population. And because they have been separated out, they are also losing out on that original genetic diversity, where the new population that they've established is not representative of the sample of the original population. So every time they reproduce, they will only carry forward the alleles that are present within that new population. And you can see how that impacts on the genetic diversity of the new population that emerges. It's a low amount of genetic diversity compared to the original population. Anyone confused? It makes sense, huh? Okay, I hope so. So why is losing genetic diversity a bad thing for the population? Essentially, genetic diversity allows for two things to be prevented. One is inbreeding. If there is a high amount of genetic diversity, then inbreeding is less likely to occur, and that means that harmful alleles are less likely to be circulated within a population. So I'm going to give you an example of that, specifically in humans as well, in a bit. Next is lower adaptive potential. So like we looked at in the previous topic, what are the reasons natural selection can occur? The four reasons. One is variability. The fact that they have variability within a population allows them to basically survive selection pressures and survive selection pressures and have a phenotype that can basically be selected for. And that's only because there was a variation existing in the population. If there is a low amount of variation, then the population suddenly become more vulnerable to new selection pressures that could potentially challenge and wipe out the entire population due to the absence of any advantageous alleles. So this ties back into natural selection, in which populations are constantly experiencing selection pressures. If there isn't a lot of variation within a population, then chances are a selection pressure could potentially challenge and wipe out the population due to the fact that there isn't an advantageous allele or a advantageous phenotype present within that population. And that is directly tied to genetic diversity. There can only exist high amount of genetic variation if there is high amount of genetic diversity. They're basically the same thing. You don't really need to interchange them that much, but yeah, genetic diversity is directly tied to genetic variation, so that if there is a low amount of genetic diversity, there's a low amount of genetic variation. That means that they can potentially be selected against as a whole, and that can lead to the extinction of the entire population. How do I put this into an easily understood way, right? So if an entire population is if an entire population of giraffe is having short necks and none of them have long necks, but all the trees are suddenly too tall, then the entire population is at risk of dying out because they have suddenly faced a selection pressure that is selecting against the entire population. But if there was a high amount of genetic variation where there were some giraffes with short necks, some giraffes with medium necks, and some with long necks, then the selection pressure would basically select for the long necks and the population can potentially survive. So only through genetic diversity can selection pressures be survived where a selected advantageous phenotype can be selected for. If there isn't a lot of genetic variation within a population, then there can be a selection pressure that can challenge and potentially wipe out the entire population due to the fact that that population has a no advantageous phenotypes to select for. Okay, this requires more a bit more example, right? So a population that is usually considered to have a low amount of genetic variation within humans is specifically older Amish populations. Because old Amish populations basically they segregate themselves from the huge portion of human populations, and they want to basically live their lives on their own, right? So from a few dozen colonists, sorry, the I mean, older, sorry, old order Amish populations are derived from a few dozen colonists who escaped religious persecution in Germany in 1719 to settle in Pennsylvania. And like I said, this population is closed, right? They don't want to mix with other populations readily, and they're not really they're not really open to the idea. They usually try to remain close to themselves. So that means what's happening? They are basically creating a system to increase their inbreeding and also reduce genetic diversity because they are only sticking to their own population. They're not allowing themselves to migrate, immigrate. They're not really experiencing any changes to their allele frequencies. It's staying pretty consistent. So what happens? Alleles and genetic disease frequencies in Amish are significantly different from the German ancestral and surrounding local populations. So how can this be a bad thing? Oh no. So basically what happens, high amount of inbreeding will occur within these populations, and whatever disease can be found within these populations are going to be passed on more and more to the next generation. So it has been found that within Amish populations, there are certain diseases that still haven't been discovered in any other population, because what happens when a specific population is inbreeding a lot is that they are going to pass down harmful alleles within that population. And that's why inbreeding is considered as a bad thing, is because when you inbreed between the same population, between the same families slash populations, you are going to be passing down harmful alleles within that population. And those harmful alleles can sometimes give a phenotype that can have a dangerous effect on the organism. So let me show you an example of that. Hmm. Yeah, can I get you guys to read this very quickly? I hope everyone can read that. No? Okay, I'll include this into the slides as well. Right. So basically, this is the part that you should look at, right? That children have medical conditions so rare, doctors don't have names for them yet, such as reported by that person. The Amish make up only 10% of the population in Ohio, but they're half of the special needs cases. 3 of the 5 Miller children, basically children from that family, have, for example, have a mysterious crippling disease that has no name and no known cure. So essentially, due to the fact that they have been inbreeding within the same population over many generations, there have been harmful disadvantages and harmful alleles that have been accumulating within that population and continuously being passed on. And over time, if it continues to happen over multiple generations, that's when you get the people who are suffering a lot of these genetic disorders because they have basically accumulated all of those harmful alleles within themselves, and that in turn gives a phenotype as a genetic disorder or a disease that has basically a very negative effect on the individual. All right, let me just include this into the PowerPoint as well. Exactly. If you look at the royal family, any example of inbreeding actually does increase genetic disorders, not because it's some kind of magic that's occurring. It's basically because, let's say, individual X and individual Y, both of them are related or both of them are from the same population that have been within that population for many generations. Individual X has a homozygous recessive gene for a harmful disease, and the same allele exists in individual Y. When both of them reproduce, suddenly you have a homozygous recessive for that gene, and that means that gene is going to get activated, meaning that disease is going to get activated, right? That's the most simplified way to think about it when it thinks about inbreeding and how it can actually emerge as a disease. Another example is actually this one right here, right? The South Atlantic island of, I can't even pronounce that, was colonized by 15 Britons in 1840. So this is a perfect example of founder effect, right? 15 Britons are gonna start a whole population, a whole new population in a isolated island. Okay, so let's say one of them is carrying an allele for retinitis pigmentosum. Yeah, I'm not going to pronounce that. So what happens is, just because one of the individuals is carrying that allele, due to the founder effect, that allele is going to be continuously passed down and it's going to become more and more prominent within the population. So among the 240 descendants living on the island today, 4 are blind by the disease and 9 others have become carriers. So that's how a founder effect can actually pass down a disadvantageous allele to a population. That's how founder's effect can actually have a very profound effect when we're thinking about a small population. So when only 15 people have isolated themselves within a population, whatever allele, disadvantageous allele that they have will most likely be passed on and become prominent within the generations that's to follow. That's because we're talking about a small population. So like I said just now, right, the effect of genetic drift is inversely related to population size. If we're talking about a large population, even if they were half of them were wiped out or half of them immigrated, half of them basically separated to form their own population, there's still enough genetic diversity that it is not going to have a high impact on the overall genetic diversity, right? But if it's a small population we're talking about, if suddenly half of them are wiped out, the amount of change in the allele frequency is going to be much more prominent, right? Here we have a population of red and normal colored penguins, and there is about 10,000 of them here versus 10 of them in this population. So one is a big population, one is a small population. Let's say we talk about genetic drift occurring, where suddenly a random occurrence has wiped out half of the population, both of them. Okay, when you wipe out half of the population for 10,000, you're still allowed to have, sorry, you're still going to remain with 5,000 of them, and in as a result, there is still going to be a high amount of genetic diversity within the alleles found within that population. But when we're talking about a small population, if we wipe out half of them, suddenly you notice they've lost one unique allele that was present within the population. They no longer have that allele. So if you look at the allele frequency for that specific allele, it has suddenly become 0%, which is a dramatic change in the allele frequency where it's lost one of the unique alleles that it originally had in that population. Whereas in this big population, that unique allele is still present, where it's basically no alleles have been lost, right? That is why I say the effect of genetic drift is very prominent in small populations and has a small effect within large populations, because large populations can basically take the hit and still be fine because the genetic diversity is still above a certain threshold. But small populations, they can't afford to take the hit, because if they do, their genetic diversity suddenly drops to a point where they could have lost potentially unique alleles that can help them survive other selection pressures later on. Any questions? It's a lot, isn't it? Yeah. Essentially, yeah. Gene flow, introduction or removal of alleles from a population due to migration, and that introduces. So if it's immigration, it's introducing genetic diversity. If it's emigration, it's reducing genetic diversity. Gene pool, remember that is the selection of genes or specific alleles within a population. When we're talking about genetic drift, that is basically changes in the population's allele frequencies due to random occurrences. Two examples of that is the bottleneck effect, where a large portion of the population died due to some random event, and thus their allele frequencies are greatly impacted moving forward. The founder effect is also an example of genetic drift, where there's a change in the population allele frequencies due to a certain portion of the original population separating out, forming their own population, and every generation that emerges from that new population will basically be impacted by that smaller population. If you, overall looked at each of them and how they impacted on genetic diversity, only two would increase genetic diversity, which is immigration and interbreeding. Immigration, founder's effect, and bottleneck effect are known to decrease genetic diversity. So reducing genetic diversity is a bad thing. Increasing it is a good thing. Why? By reducing genetic diversity, you're increasing the chance of interbreeding, and you're also lowering their adaptive survivability, where they are less likely to be able to survive new selection pressures that they are going to be facing. And that can in turn impact the survivability of the overall population as a whole. Right? And that makes the four mechanisms of evolution that can change the allele frequencies of a population. First one is mutation, where there's a random permanent change within the DNA sequence of an organism, changing its phenotypes, changing its genotype and expressing a different genotype, and that's specifically if it's happening at a coding sequence. Natural selection, that's when a selection pressure is actively selecting against a trait and selecting for a trait, allowing that trait that is selecting for to be able to pass on its advantageous alleles to the next generation, meaning that that trait that is being selected for will become more prominent in terms of its alleles in the next generation. Genetic drift, that's when a chance event has suddenly changed the allele frequencies of a population, where it's either going to kill off a huge chunk of a population, allowing the remaining population to emerge with a reduced amount of genetic diversity, or a small population suddenly emerges out of that original population due to a chance event, causing that new population to greatly impact on its genetic diversity and allele frequencies. So individually, when you look at them, they're easy to understand. Overall, when you try to relate all of them together and link lines together, it kind of gets a bit more complicated. You can probably get a better understanding of them if you do more questions relating to them, right? All right, let's look at this one. Consider the diagram below the gene pool of a population of over 20 generations. So notice how the population has changed over the multiple generations. It would be correct to conclude that over 20 generations, what has happened? This is one of those questions where you don't really have to overthink it, just process of elimination. Go through the answers, see which ones make sense. All right, I hope everyone has an answer. Let's go through a discussion style, okay? Gavin, do you think A makes sense? Do you think genetic diversity is increasing over the generations? No, right? So A doesn't make sense. Let's kick it out. Now let's see. Yeah, put. Does B make sense? Individuals with the genotype homozygous dominant RR have a selective advantage in this population. Does it look like that's the case? No. So let's kick it out. All right, now let's see. 10. The C makes sense. The frequency of each allele is equal in generation 1, but not in other generations. Does that make sense? Maybe. So let's keep it under advisement. And D, can I get Ayman? The new advantageous alleles for this gene were introduced as individuals joined this population. No, there are no new alleles, right? They're all the same alleles that existed in the original population. So the most appropriate answer is C, right? So this is how you use the process of elimination in answering questions. You should be able to do this very quickly as well. And that would help you out